Hey guys, welcome back to the AS Biology in 1 month series. And yeah, I've completely finished. I managed to get my results. So here was my exam paper. And as you can see, I managed to scrape 73%, which was an A star. As you can see, A star grade boundary at 68%, cohort average at 65%. So we've managed to scrape this A star grade boundary in 19 days. And in this video, I'm just going to be going over the paper. I will say I was not doing too well in this paper. Not sure what happened, but I guess a lot of the questions were a bit different from usual, so it kind of threw me off, but still managed to scrape this boundary nonetheless. But yeah, I'm just going to be going over my mistakes. The paper, I've got the mark scheme on the right here and the actual paper on the left. And yeah, I'm just going to be going over seeing what's happening. So yeah, I'll make a video on the exact steps you need to follow if you also want to get an A star in 19 days, but this video is more like a reaction sort of video. So yeah, let's just get started. First question is about a human heart, and okay, so this was alright. Like, I managed to secure these marks. I'm actually writing a lot for just 1 mark, but you know, it's biology, so what can you do? Essentially classic left and right side. And yeah, just have a look at the mark scheme. You can see separate oxygen, separate deoxygenated and oxygenated blood. And yeah, so this was all fine. I think the mark scheme had some special words circled, but that's alright. And alright, so first few questions are all good. This heart stuff, all nice and easy. Covered that quite a lot. Switch blood vessel, vena cava. Going over here, you can just see I've sort of labelled the left ventricle and just secured all of these marks. Again, I've seen a similar question to this, so I knew that blood vessel R was actually the aorta and blood vessel S was the pulmonary vein. So now I can just talk about differences between a vein and artery and the aorta in particular. So here we go. Let's move on. So yeah, this was the first horrible question. We had a question. The question was about a heart of an insect. You can see the diagram here, and it basically asked us to compare and contrast the heart of this insect with a human heart, which was seen over here. So in hindsight, I suppose I definitely could have been a bit more specific. Just name the features of a human heart and say they're not present or present here. But I think in the exam I was just thrown off, given this is a completely different sort of system. And yeah, so if we have a look at the mark scheme, you can see both have hearts and both have valves. Yeah, that makes sense. Should have said that. Mammals have a closed system, separate pulmonary. So yeah, so mammals have a separate pulmonary system. I can't remember if I actually said that one. I obviously didn't, but yeah, I just really wasn't doing too well. Double circulatory system, whereas insects is single. Yeah. Oh, right. So I've actually talked about that here, but I suppose I wasn't specific enough. Alright, yeah, that's quite weird. I feel like I should have got an extra mark there, but it's alright. No point asking now, the exam's finished. So, weird question number 1, but managed to get at least 1 mark. And then here, carried in a hemolymph, so I kind of saw hemolymph coming out of the blood. Thought this would be similar to plasma in human blood, but turns out evidently oxygen was not carried, which I guess makes sense. I think I was thinking of hemoglobin, but probably should have thought of more sort of plasma oriented things. So you can see glucose, amino acids, basically everything that's carried in plasma were answers here. And okay, I suppose you can see for the growth, so that kind of gives it away. First question we get 9 out of 13. Now number 2 is a phospholipids question, and I think I'd seen a question very similar to this beforehand. See, I even drew this diagram. Yeah, so this was an easy question. Just had to talk about the hydrophilic and hydrophobic parts of the phospholipid, and then talk about the bilayer. Again, I've seen a similar question. I believe this was one of the EdXL questions, so I'd probably seen it, or a variation very similar. So I knew movement on water by osmosis. It is down a water concentration gradient, which is the same thing as against the solute concentration gradient. And yeah, I was able to secure full marks, I believe, on this question. So yeah, that was quite good. It's relatively easy, just simple stuff. Got the carrier proteins here. Uptake here tells you the base there, so you could work that out. And then I had seen a similar question to this CTFR protein. Also done a lot of work on it. Once you understand it, it makes a lot of sense. And you can see I actually wrote so much. Finished this, then went down here and carried on. That's fine. Yeah, here's the work here. So you have to talk about chlorine, then sodium, which I believe I did. Yep, here. And then we just talk about concentration gradient increase in osmosis. Got the marks there, and then same thing here. 3 things you really need to talk about with the genetic code are the triplet code, so codons forming triplets, the degenerate nature, and the non-overlapping codes. And I was able to just show that in 3 different bullet points, nice and easy. Yeah. Also notice how they've underlined certain words, so they're actually making the mark scheme harder, but luckily I was able to capitalize on that. Again, I'm fairly sure this is an EdXL question, so I definitely seen it. I definitely remember the elephants. So once I knew that, it was really just first sort of calculation question, nice and easy, just a Hardy-Weinberg. Plug the numbers in and substitute. Then we have here, and so yeah, I've actually not managed to get this mark. This was a natural selection question, but it was asking why the selection pressure was the fact that they had tusks, so relatively simple there. But I haven't actually managed to get these 2 marks. Let's see what's going on here. Calculate the allele frequency. That's weird. I have said this. Calculate the allele frequencies. Right, okay, I understand. So it's not enough to just say calculate. I suppose you have to say how it can actually calculate rather than just calculate. That's fine. And yeah, I guess I had to say sample in a specific time, so they've definitely made that question a lot harder than it should have been. I have basically said what's on the mark scheme, but I guess they were looking for something a bit more specific. It's alright. Yeah, okay, that's fine. Maybe I'll tally up how many marks I think I should have got. Not like I'm really gonna ask them to remark it anyway, but I guess it's just interesting. So let's write this down. Alright, yeah, so should have got 1 on question 1D and question 2C, 2BII. Yeah, let's just keep a note of this stuff. Moving on to question, this is question 3. Oops. Yeah, moving on to question 4. Again, I'm fairly sure this was an EdXL question, although this is actually a bit hard to remember. Relatively easy though. I remember having to talk about undifferentiated and the ability to differentiate for the 2 marks here, which saved me. Going on here, got benefit of a doubt there. And okay, so yeah, this was a self-specialization question. Sometimes these are 5 marks, but I guess this one was only 3 marks, which is fine. I should just describe the process of specialization there, and then epigenetic changes. So talk about histone protein modification, DNA methylation, acetation, and the effects they have on RNA transcription. Then just talk about protein synthesis and how it can affect the phenotype and genotype, or gene expression. So managed to get those marks. Moving down. Yeah, this was easy. Hadn't seen this question before, but it's alright. yeah, you can see I've completely fumbled this. I remember being really confused what a slime capsule was. Then I remembered it was just a capsule around bacteria, but I still couldn't quite remember the function of it. I suppose it's for protection, right? 5A. This must be a misprint. So 5A prevents the dehydration of cells. Alright. I guess I should have said that. I mean, I said it acts as a protective layer, but I'm not sure that was enough, really. Yeah, and then I had absolutely no idea what Pili was doing. I guess it's for adhesion. Alright. Yeah, that's good. Right. Moving on, we have this 6 marker, and I only got 3, which was weird. It was talking about different methods, so I talked about the 3 stages of clinical trials, then talked about double-blind placebos, different sampling methods. I suppose. Oh, okay. So they're giving me 5, but then only give me 3 here. That's pretty odd. I wonder how many marks I actually got. It's out of 10, so I should have lost 2. Oh, okay, yeah, so I got 5 out of 10, right? Yeah, makes sense. But let's have a look at the mark scheme. I wonder what this wants. So, okay, so we have to talk about. Okay, yeah, that's optional. Testing on healthy people. Right, okay. I might have just said ill. Talk about group sizes here. Test. Okay, statistical test, that's important. Place randomly. Yeah, so I've done most of this, but I guess I just. Huh. That's weird. Yeah, I definitely feel like this was marked kind of harshly. Because I'm fairly certain I've talked about essentially all of these, but I guess I haven't talked about group sizes, so it wasn't that detailed. Yeah, so phase 3 is test on a much larger group once the confidence has been built up. Alright, yeah. Maybe I'll petition that. This is a 6 marker, so it's kind of hard to petition, but you know, I guess this is the answer. Just make sure you talk about group sizes. Scribbled, underlined this, so maybe they're trying to say something. Yeah, alright, that's fine. It continues on the back. We'll go to that in a second. Okay, so I'm actually quite confused why this hasn't been given a mark. An organ is made up of different tissues. Alright. Yeah, okay. Got a bit confused. I remember you had to say something about same function, shared function, but I suppose I got it confused. The cells, the different tissues work together to perform a shared function. Alright, yeah, that's fine. And don't know what was going on here. I think I drew anaphase by accident, then I realized it said metaphase, so I changed it quickly. I still only got 1 mark though, which is weird. Let's go to the back and see if they looked at it. Okay, so they crossed out centromere. That's weird. I have labeled the centromere. Like, was it not clear enough? Okay, I'm gonna put that up for petition since I feel like that it should be given a mark. That's right. And okay, yeah, so you can see the continuation of 5AII. Yeah, I guess it just wasn't enough to secure more than level 2. Unfortunate. Anyways, let's carry on. Describes the event, so got those 2 marks. That was GCSE level question. They line up along the equator and spindle fibers attach. Then, oh, I've only got 2 marks here. Right. First mark was for, alright, I didn't get mark phase 4 or mark phase what? Mark point 1. Number of cells in metaphase increases because cold chain stops the cells from. I believe I did say they're stuck in metaphase. Yeah, stuck in metaphase over here. Suppose they didn't give me that mark for that one. And then I didn't get mark 4 either. Number of cells in prophase decreases, fewer cells. Again, I'm fairly certain I've said all these marking points, but maybe not. Okay, we'll let that one slide. Describe how these, yeah, this was a simple one. Basically described a root tip squash experiment, but just explained it on animal cells rather than plant cells. Got the marks there, and yeah, okay, managed to get all of them there. That's fine. So yeah, here was another Edexcel A question. I remember this one. I'm actually lucky I remembered it because this is a really confusing question, but I just remembered to look for the large one, and that was the xylem, because I'm going to reduce the resistance. I could also figure out that was a phloem. I think it's a bit clearer on the actual paper, but that's like a sort of tube, like a garbage duct. Just how I remember it. But yeah, once I got that, it was easy. Xylem, phloem, and also had to remember this. So sclerenchyme was on the outside, then phloem, xylem, kind of like over here. So I got all these multiple choice. That was fine. And made a silly mistake here. I measured this distance as 3.5 centimeters when it was actually 3.4 centimeters. So that was silly, but it's fine. I only lost a mark there. Like I said, I wasn't doing too good in this exam, so you know, this was really annoying because I knew these mark points like on the back of my hand, but I guess I wasn't specific enough. I actually questioned my teacher. She told me here the use of fibers from nettles will be available to future generations, and I wasn't able to get that mark even though I said available for future generations, because I talked about nettles rather than the fibers from the nettles. So that was annoying, but it's alright. I guess you just have to pay more attention to those. Also didn't get the 4th mark. I don't think I said biodegradable. Oh, I did actually. More nettle fiber. But this time you had to say the clothing was biodegradable rather than the nettle fibers. So, you know, it's alright. That is why biology mark scheme is so notorious. This was a weird question. Wasn't sure what to write, so I said people underestimate, but then didn't know how to explain this one. General. And so I suppose this you have to say, since they're relatively similar, they have a realistic perception of the risk rather than overestimate. Could have said that for smoking as well. That's fine. And yeah, it specifically says ignore perceived risk is lower. Oh, wait, okay, hang on. Accept perceived risk. Right, okay, yeah. So that was just unfortunate. It was a bit hard to like explain, but so much to get a mark there, and oh, completely lost all the marks here. Alright, what's going on? So one marking point was a large sample size. I do not think I said that. I really mostly talk about control. Should not know. No known health risks. People knowledge. So yeah, you should control the knowledge. You should control the people themselves, or not them, not the conditions, but they should have no. They should have no knowledge of their health conditions that might skew results. yeah, I suppose just make sure you talk about large sample size for these kind of questions. Only I lost another 2 marks there, which is so annoying. I will say, I feel like this paper's been quite harshly marked, but might just be me. Explain why it's important that person's perception of risk. So I tried to explain it here. I suppose I didn't hit the mark scheme points. Lifestyle can be modified. So yeah, it's really talk about the treatments here rather than the actual problem. So if you properly know your actual risk accurately, then you can accurately modify your lifestyle. You can get medication, and I managed to secure mark point 4. Okay, yeah. So I have to just explain how these medications could be given effectively if the perception of risk is on point. Alright, now I think this was one of the last questions. This was a little diagram and then talk about how to use it. Ooh, alright. This is quite annoying. I guess it says explain, so stop smoking and then explain how smoking. Yeah, okay. Stop smoking, this would reduce the points. Yeah, okay, so I need to explain how quitting smoking would do the changes. Decrease higher intake of saturated fats. Yeah, so I feel like I could have definitely got much higher in this paper. Like, I feel like I could have gotten the 90s if I just sort of hit the marking points better, been more on point in the exam, because I knew what was going on here, but I suppose I didn't explain it well enough and I didn't read the question. So maybe I'll try again later. Who knows? Maybe I'll try and get 90%. Again, you can see I've told her to reduce her intake of saturated fats. I don't know if that was anywhere, but yeah, okay. Yeah, okay, so I didn't explain how it leads to atherosclerosis. Alright, that's fine. Just losing marks everywhere. And suggest why this method might not be accurate. Let's have a look. So I haven't included other risk factors. Cholesterol might have to be estimated, and alright. Yeah, so there you have it. Let me know if you think there was a bit of if you think there's anything wrong with this marking. Honestly, I probably not gonna petition my marks, but I will say I think I definitely had potential to get 90% or at least 85% in this paper, because I've just been making so many silly mistakes. It's alright though, still got the A star, which is the most important thing. But yeah, that's this paper complete. Overall, managed to get an A star, 19 days of revision. So I'll give you a tutorial on how that will be accomplished soon. Thanks for watching, and I'll see you guys in a future video. Bye.